If a client has poor glucose regulation and significant digestive symptoms, you don't need to necessarily look at these as two completely separate problems. It could be related to stress, food quality, sleep, training, so many other factors. So insulin resistance, dietary quality, energy balance, body composition, alcohol intake, and micronutrient adequacy all are part of this person's metabolic status quo and matter in terms of this conversation around gut permeability and hyperglycemia. Welcome to Sammillerscience, where you're joining me nearly 2 decades into my personal journey to bridge the gap between macros, metabolism, muscle, and medicine. If you're joining me, thanks for trusting me to elevate your health and realize your true potential. Each week and inside every episode, I'll be sharing methods and models from my coaching journey and my personal experience, as well as the science that you need to achieve whatever specific goals you're working on in your life. Let's jump in to today's show. Could high blood sugar be disrupting the gut barrier and raising infection risk? In 69,000 people with type 2 diabetes in northern Denmark, they were tracked through national health registries for over a decade. And then researchers looked at their A1C values, which is a measure of insulin sensitivity in blood work, and then they compared that to infection records side by side. The people whose A1C ran the highest over time were 64% more likely to end up hospitalized with an infection than the people whose A1C stayed in the low sixes. And for infections treated at home with a prescription, the increase was 19%. Now that relationship has been under our noses for years. This research came out around 8 years ago. And the usual explanation is that high blood sugar impairs immune function. But recently, a group of researchers at the Weizmann Institute went looking for what else could possibly going on beneath the surface and explored the specific relationship between high blood sugar and intestinal permeability, which is also known as leaky gut. The first paper was published in Science in March of 2018 and explored how hyperglycemia drives intestinal barrier dysfunction and risk for enteric infection. I'm covering this because a lot of you listening could either be experiencing this yourself and maybe have some gut issues and high blood sugar, or you have a client right now with fasting glucose in the low hundreds, an A1C that wasn't addressed by Western medicine or their primary care doctor, and gut complaints that top it off. Now, the statistics from the Danish study are really interesting because in every person that had type 2 diabetes was basically diagnosed between 2000 and 2012, and then the researchers had access to both their lab values and their prescription and hospitalization records. So this is a form of observational research. Then they compared the highest A1C band to the controlled band, and hospital-treated infections came in at a risk ratio of 1.64 and community-treated infections at 1.19. When they used a single A1C taken at the time of diagnosis, that relationship almost disappeared, and the risk ratio for community infections was 0.97. The indication only showed up when they used the updated time-weighted A1C. And this is really the part we want to pay attention to, is more the blood sugar aspect versus hyperfixating on the infection risk ratios. So this means the cumulative glycemic exposure across the whole follow-up period. So the trajectory carried what a specific snapshot missed. And we're going to cover that more as this episode unfolds. So inside the second study, which is the more recent paper, the authors recruited 27 healthy volunteers and ran a full serum panel on them. They also had different metrics like BMI, waist circumference, and they had blood pressure, their lipids, liver enzymes, and A1C. Then they measured how much bacterial material was circulating in some of these blood samples. Hey everyone, if you're listening to today's episode or you regularly find value in the content, if it's educational, entertaining, or changes your perspective about certain health topics, the greatest compliment that you could give my team and I is a rating and review. Also sharing the show with any friends or like-minded potential listeners who might benefit from the content that we share here. My team and I invest loads of resources into producing this production for you every single month without the influence of outside sponsors who potentially could negatively influence the education and type of content that comes straight from me to you. A lot of podcasts in the health and fitness space, every 5 minutes you're having to hear about a different product or commercial or supplement that you're supposed to buy or something you need to put in your house. And that's not the way that it is here. My team and I bring you the latest and most relevant health information to support you as you work towards your goals. Or if you're someone who's a health and fitness professional and you coach others, you can take this directly back to your business for clinical application or even in the content that you share. So help us continue the ripple effect of this health information by sharing it and supporting it with that rating and review. Without further ado, let's jump back into today's podcast. Of every variable on that panel, A1C had the strongest relationship with circulating microbial products. The correlation with TLR4 ligands specifically landed at a p-value of 0.0081. BMI did not reach significance, and neither did the other standard markers of metabolic disease. The A1C range across those 27 people ran roughly 4.5 to 5.5%, which means every one of them would have been told that their blood sugar was in the normal range as far as Western medicine goes. Now, the mice study exists to answer the question that human correlations cannot, which is whether the glucose is causing this issue of gut barrier dysfunction or just happening alongside of it. Now, 4 findings from this study and the animal side of the research tell us what we need to know paired with the human evidence. The first is that they separated glucose from obesity. So diabetic mice with a leptin receptor defect showed a compromised intestinal barrier, measurable as reduced tight junction protein, increased passage of large molecules into the blood, and collapsed electrical resistance across the epithelium. When infected with intestinal pathogen, they could not contain it and the bacteria spread to their liver and spleen. Then the researchers restricted those mice to the food intake of their healthy littermates until their body weight normalized, and the failure persisted. They also ruled out the immune system by swapping bone marrow between healthy and diabetic animals and finding that susceptibility followed the host tissue rather than the donated immune cells. Now, these are things you can only do in rodent studies, not in humans, so that's why we're using the animal evidence here. The second is that they reproduced it in lean animals. 2 different models of type 1 diabetes, one chemical and one genetic, neither involving obesity, produced the same barrier breakdown and the same systemic spread. The third is that this issue reverses. Insulin at a quarter unit per day for 4 weeks brought glucose back to normal in those animals, and the gut barrier came back with it. The fourth is the mechanism itself. So glucose actually enters the intestinal epithelial cell from the bloodstream through a transporter called GLUT2 or GLUT2. Inside the cell, it then reprograms metabolic gene expression, and one of the pathways that is hit hardest is N-glycan biosynthesis, which is what allows junctional proteins to be assembled and trafficked into place correctly. When the researchers removed this GLUT2 from intestinal epithelial cells only, those mice kept their tight junctions, they kept the bacterial products contained, and resisted the infection while their blood glucose stayed high. They also closed the door on the 2 explanations that our industry in health and fitness probably would have reached for first. So they transferred the altered microbiome from hyperglycemic mice into germ-free animals, and it did not transfer the barrier problem. So dysbiosis was not the main driver here. And the glucose effect showed up in cultured human intestinal epithelial cells with no microbiome, no immune system, and no food anywhere in the system, degrading tight junction architecture in a dose-dependent way. So the translation to actual humans and what might happen in clinical practice here is that intestinal permeability is interconnected with glycemic status, and it's something where we saw as hyperglycemia worsened, it made the barrier function or intestinal permeability worse. The connection between high blood sugar and intestinal permeability, or the leaky gut language that your clients probably use after hearing about it on social media, is best understood as a bidirectional relationship involving glucose regulation, inflammation, insulin resistance, and gut barrier integrity. So chronically elevated blood glucose can increase advanced glycation end products, or AGE. So this formation is happening with that elevated blood sugar. It also can contribute to tissue stress and greater systemic inflammatory burden. That's important because intestinal lining is a selectively permeable barrier. Maintaining that barrier requires healthy intestinal tissue and appropriate immune regulation. So in a chronically inflammatory metabolic environment, maintaining and repairing the intestinal barrier becomes more difficult. So conceptually, the way we want to think about this is that chronically elevated glucose then is contributing to AGE formation or advanced glycation end products, and that's leading to tissue stress. There's greater inflammatory burden downstream of that, and then reduced gut barrier resilience is happening as those things are taking place. That reduced gut barrier resilience leads to increased intestinal permeability, and when intestinal permeability increases, the barrier has a harder time controlling what crosses from the gut into circulation. Remember, the gut is selectively permeable, and then bacterial endotoxins and other luminal material can have greater interaction with the systemic immune system. That can stimulate additional immune and inflammatory signaling. So when you have that, what's happening here is increased intestinal permeability, or leaky gut, leads to greater exposure to bacterial endotoxins, and then immune activation follows, which then triggers greater systemic inflammation inflammation. This is where the relationship can become circular though. So blood sugar and gut health are really interconnected in a way where we can't separate them. So chronic systemic inflammation can then contribute to insulin resistance. So now this is a feed forward cycle. It's being like stuck on a hamster wheel going in the wrong direction. As insulin sensitivity declines, the body needs more insulin to appropriately manage glucose. And as that process progresses, glucose regulation can deteriorate further. So rather than thinking in a straight line of high blood sugar directly to leaky gut, it's more accurate to think in terms of high blood sugar and elevated insulin leading to or contributing to inflammatory burden and impaired gut barrier integrity. This also contributes to greater exposure to gut-derived inflammatory material. It also can increase or lead to additional systemic inflammation, the worsening of insulin sensitivity, and poor glucose regulation as a result of this. So this is kind of the full cycle rearing its head. That creates the potential for a self-reinforcing poor metabolic health loop or a poor gut health loop. So all of these things lead to an increased inflammatory environment and make gut health harder to maintain. And each one of these issues, the blood sugar issue and the gut issue, are now reinforcing each other. Following today's show on leaky gut and insulin resistance requires the ability to hold space for 4 different bodily systems or conversation around 4 different aspects of science: immune function, microbiome, insulin resistance. And that can feel like connecting the dots or trying to put together puzzle pieces that seem separate and sometimes confusing at times. And a lot of health coaches struggle to apply that science to real client transformation. But that's exactly what we do inside the Functional Nutrition and Metabolism Specialization Program. You'll work live with me and my team on real cases alongside other coaches and practitioners who are either running into these problems or want to be prepared to handle them when these cases show up on their doorstep. You can learn more in the show notes or head over to metabolismschool.com to learn more and apply. Now, we're going to go through some of the precise evidence on this and what the evidence actually supports in terms of the difference in the human and animal data. Now, the causal mechanism is established in mice and is demonstrated on human intestinal cells in culture, but that's different, right, than having an RCT in humans, but it's hard to have an actual treatment group on this. So the link between glycemic control and circulating bacterial products rests on a cross-sectional sample of 27 people. And the clinical outcome, more infections at a higher cumulative A1C, comes from large populations with diagnosed diabetes. Nobody has run the study that connects those 3 tiers in a living person, which would mean improving glycemic control in humans and measuring intestinal permeability and infection outcomes as that insulin sensitivity improves. So the claim that you can defend is that glycemic status and intestinal barrier function are mechanistically linked with strong preclinical causal evidence and consistent human association. Now that's human association, not causation. So what you can't say here is that lowering a specific client's A1C or fasting insulin will automatically resolve their specific gut symptoms. And the distinction is one that is pretty important, and we'll see as the research and evidence continues to unfold. So if a client has poor glucose regulation and significant digestive symptoms, you don't need to necessarily look at these as 2 completely separate problems. It could be related to stress, food quality, sleep, training, and so many other factors. So insulin resistance, dietary quality, energy balance, body composition, alcohol intake, and micronutrient adequacy all are part of this person's metabolic status quo and matter in terms of this conversation around gut permeability and hyperglycemia. Do not infer hyperglycemia from digestive symptoms alone. So blood work, fasting glucose, fasting insulin, A1C, triglycerides, and HDL interpreted together rather than relying on a single marker can provide a much clearer picture of blood sugar regulation and insulin sensitivity. The key concept is that gut barrier function can increase inflammatory burden, while metabolic dysfunction can create an inflammatory environment that makes good gut health harder to maintain. Each can potentially reinforce the other in a subset of clients, depending on their background and health history. I created today's episode because blood sugar has lived in a separate bucket inside the health and fitness industry that's often spoken to completely separate from gut health in most conversations. So you hear people talking about dysbiosis or intestinal permeability, and rarely is there a mention of hyperglycemia in that. So we need to be able to connect the two and understand that there's overlapping issues that are impacting clients in their daily lives. If you enjoyed today's episode, learned something new, or if this changed your perspective, please share the show. It only takes 2 seconds to support the content that my team and I share here, and it could be as quick as just dropping a 5-star rating wherever you listen. We had over 300,000 streams last month combined between platforms, and over 80% of you guys aren't subscribed. So smash the subscribe button and make sure you never miss an episode. That way you get first dibs on all our latest research and metabolic health updates. Thanks so much for watching, and I'll see you guys in the next show.